Many programs, and it's interesting to see how many people have already come quite far in their program. Also, what you see now and then is that there is a lot of contact with AZA. Also, AZA, the American association, is having a lot of programs in, on elasmobranchs. And why do everything by your own when you can collaborate? And there is a lot of collaboration between different programs on the same species, so that's really, really great. I'm not Nuria, but Nuria could not be here. So I just give a short overview of the ESB on Tenura lima from Nuria in Lisbon. She started in 2007 and published already 2 stud book overviews. And you can see that in the second stud book the number of animals is still increasing, but also the number of institutions is increasing. And this is not only because there are new institutions getting this species, but also because there are more and more people of you starting to send in their data. So it's really great to see this. This is just the current living population. 22 males, 38 females, and yeah, it looks really promising. You see the increase of the living population over the years. Many of these graphs, they start in end 90s. That doesn't say that this species was not already there before in Europe, but again, this is a lot of registration. Many people don't know anymore when exactly were the animals there, and yeah, from 1998, more and more people, more and more aquaria were starting to register their animals, and so they can give knowledge from historic data. Captive birth in this species is really good. You see more and more institutions being successful. 10 in 2012 and 8 animals in 2013, so that's really promising. And I hope, of course, that this will proceed. 32% of the living animals are captive bred. So in that perspective, the sustainability of the whole population is getting better and better. There are different transfers in 2013, and we hope to have more in the coming futures. There are 14 institutions that are having population or potential breeding pairs, so hopefully more and more births will come up. Nuria wants to do a lot in the future, so she wants to increase further the number of participants, and because you have so much offspring, new couples can be formed, especially when more institutions are breeding, so you can have genetic independent couples formed, so this way you get also new participants. But potential breeding pairs, of course, is one of the major things. You need to have a couple to be able to breed. And of course, increase in knowledge will be a constant issue to work on. Husbandry manual. Well, it's really good to see that the manual, the name manual, is going into husbandry note, and I think that will make it much easier for any coordinator to start to work something out and just to have a note only on the breeding or only on the feeding or only on the health, and that makes it's much more workable. Genetic analysis is also important on the population. Please contact Nuria if you're not on the list yet. Nuria is also a coordinator of the Sphena levini. Yesterday we saw some very nice animals swimming around, and it's a species which is quite new in Europe. This monitoring program just started in 2013 because of the RCP workshop in London. She wants to follow the trend, what is happening in Europe, what problems do people have with the species. It's not the most easiest species to keep, and there are different issues to tackle. And hopefully, when combining all this information, you will get more and more information on how to keep these species. And when you compile all this, everybody can learn from it, because that's, of course, really important. The current living population, is already 37 animals in Europe, so it's quite a large number. And that's also because in the last 2-3 years, most came into Europe. Again, please contact Nuria if you're not on the list, and yeah, she can ask you further on this species. Also, when you're interested in the species, in whatever species, please try to check the coordinator. Jean-Philippe Cateau, he's the stud bookkeeper of Carcharinus plumbeus. This is a big stud book. It's a lot of work for him. And I just phoned Jean-Philippe last week, and he was saying, yeah, it's a lot of work. And I really need to have a little bit help. So if anybody is interested to help Jean-Philippe further, bringing the stud book another step further, that would be really great. Please contact me or Jean-Philippe directly. This is a stud book which was improved in 2010, and there is a lot happening with that species. There are not an enormous number of participants, but still there are quite a large number of animals. And I think the number of participants is still increasing. This is the age pyramid of the animals within the population, and it's quite interesting to see that yeah, the oldest is 40 years old, 40 years in captivity. So I think it's really good to be able to analyze such data from the population. This is quite an extensive list. But you can see that, especially on the right side, where are we here? Living Pup, there are 4 institutions that are very successful at the moment in producing young, and that's really beautiful to see. It's a big shark species, and that you are able to breed, and that there are more institutions also able to breed. That's really promising. And of course, Antibes is incredible what they do. 200 young. So it's incredible how many young they have had. Very successful. The ESB for the stegostome, for the zebra shark, started in 2007. There are now already 40 participating public aquaria. It's a very nice species, and we work a lot together with the AZA, with Lice Watson. This is an old name I see, Lice Watson. At the moment I'm working on a husbandry paper for the Monterey Bay Elasmobranch meeting, together with Lice, on different issues of husbandry of zebra shark. The captive population also increased a lot over the years. But what's really interesting to see is that there is also a lot of birth coming up, and not only one institution, but there are now already 3 institutions in the last year that produce young, and that's really nice to see. Not all young are still alive, so there are still a few challenges to cope with when you have young, and also there are more and more participants mailing me that they have eggs now of the species. So it's really nice to see the development of this population. And it's not because it's an ESB, but it's also because animals are getting older and couples are formed, and I think also the whole knowledge of this species is increasing. Something I would like to mention shortly is that there were different breeding loan transfers. Breeding loan is a very common activity within the zoo community on mammals and on birds. You just send an animal to another aquaria or to another zoo, but it's still your own animal. So you bring them on breeding loan. So you lend them the animal. You can do that for a specific defined time, depending, for example, we sent one zebra shark to Hamburg, and together with Guido we discussed how long should we do that. Well, we decided to do it around half a year, and then after half a year the animal would come back. But you can also do a breeding loan and just give the animal to, for example, to Hamburg and say, okay, this is your animal. You can keep it for as long as you want, unless you have young. Because maybe when you have young, it would be nice to have also some young back. Or maybe in the future, I would like to have the animal back. And so with breeding loan, you can make a contract together, and you can put in whatever you want, and you can both agree on that. And it's very nice to see that there were 3 different breeding loan transfers with this species in the last couple of years, and that's something which is coming up. They have not been, all 3 have not been leading to a successful young, although the last transfer from Berlin to Hamburg, I believe there were some vital eggs already, some live eggs. So in that perspective, I think it was a big step. Maybe also nice to say shortly is that in Arnhem in 2012 we did an artificial insemination on Stegostoma. This was fun. This was so much fun that we really didn't know what we were going to do. So together with the vet we were looking how should we get the sperm out and how can we bring the sperm in. This is something really new. We decided to do it again in 2014. But this time we also try to get some more people involved. Yeah, we are not looking too stupid in how should we do this. So it's really a trial and error. I've been in contact with already other aquaria that are also trying this. And I hear from more species of Elasmobranchs, they are trying artificial insemination. So maybe that will be an interesting step for the future. But I stepped down for this stud book. There will be 2 new stud book keepers for the zebra shark. That's Sylvia and Katie. I picked up the co-chair on the FAITAG, and yeah, there's always a limited amount of time. So I'm really happy that 2, Katie and Sylvia, want to pick up the stud book and bring it further. So I'm out. So if there is any question or anything happening with your animal, please contact them. And well, I'm sure, I'm really sure that this stud book will be put further into new big steps. So it's very nice. The last overview is on the white-spotted eagle ray stud book. This is also what I'm running at the moment. We do that now since 2010. 12 participating public aquaria. If you look at the historic population, again, it starts somewhere in the 90s. There was one animal mentioned in Berlin in 88, and then you made a big step to 1998, 1997 to see that there were some animals. Well, there is an enormous increase in this species within Europe. There's a lot of interest in the larger aquaria. Also, the breeding is going quite nicely. In Arnhem, we have now 30 young in total. But also Dubai is starting to produce young, really nice to see. The only problem is when you breed is that you don't know who daddy is. So we did some research. For the female you can see it, also in Copenhagen you could see there was one of the eagle rays swimming there with a very big belly. So I'm pretty sure she was pregnant. But when you have a group, in Arnhem we have a group of 4 females and 2 males. The female is easy to see, who is mommy, but we don't know who is the father. So we did some genetic research by picking the poisonous sting of the animal and try to analyze that and to see if we could find out who the father was. Well, both fathers were active, but it was quite nice to see that some of the females only went with this father, and one other female only went with that father, and one female thought, why not do both fathers? So yeah, that was nice to see. It was even so strange that of the lady who was going with both males, she had a twin of one male and of the other male. So within one twin she had 2 fathers. So it was quite nice. So the genetic research will help in any case better for this stud book. We are now going into another step of genetic research because the species is a bit of a mess. And you could even see it yesterday in Copenhagen. There were some white-spotted eagle rays with white spots, and there were some white-spotted eagle rays with not white spots. And yeah, you see that all over Europe, and you see it also if I just take a few pictures here from internet. There's a lot of difference, only by the look, but also by the geographic distribution. So a lot of research is done within the scientific world. Is this 1 species, or is it 3 species, or 5 species? So we are now sampling animals from the captive population within Europe and try to see if we can find out which species we have. And to be honest, I'm pretty sure now that we have not Aedobates narinari in Europe. So the stud book of Aedes narinari is from today finished. But now we need to know what other species we have. And for example, from Arnhem we have young, and we send them to other aquaria like Montpellier, where they have already some eagle rays. But is it the same species? Do you combine now the same species? The sex ratio is now good, because they only had 3 males, and now they have a female. But when it's a different species, you don't want to combine. So it's quite vital to know the genetic taxonomy. This is what I wanted to tell you about the eagle ray. I would like to have a short discussion. If there are any questions about the studbook presentation, so please can all the studbook or monitoring coordinators come forward. Piet? No.
I would just like to add a little comment to the dots on the eagle ray. That eagle ray you referred to yesterday in Copenhagen was, as when it was young, it was dotted like the other eagle rays. So I don't know, it's probably the same species anyway, but it seems that when they go in captivity for a while, they lose their dots.
Yeah, that's right. That's also what we see with the population we have. So when young are born, they have in the back, they have nice white dots, or sometimes half of them. But when they are 1 or 2 years old, the dots are gone. So yeah. Any other questions?
Regarding the manuals, AZA is kind of looking at the Elasmobranch manual and that series to kind of take the place of actually having to do the very formal and tedious manuals that the rest of the zoo people have laid out. What's the attitude of that over here in Europe?
Well, the attitude is that we try now to create husbandry notes. So not a whole guideline, but more and work towards notes. And yeah, what is a note? It's that what you think is important for your species to tell to everybody, or that you found out some interesting husbandry information to share. So that's a more easy way of combining information.
Okay.
Does that answer your question?
Yeah, partially. Also, I think those sawfish young are still alive.
Sorry?
The sawfish young are still alive, I think.
Okay.
Thank you.
Any other questions? No? Okay. Yeah.
I just have a comment there. One thing that would be nice is to be able to join all the people together who are in the studbook, so that when someone has a birth, so at the moment, hello? At the moment, when we have a birth at one of the aquariums of a blacktit, for example, it'd be very nice for all the other people that are part of the studbook to get that kind of live information, so they learn from whatever happens. And so I'm still looking for the right place to put that, whether it's an email thing that we're all on the same email and we just send out an email each time, or we have, I don't know, some other kind of IT solution. But if anyone else has already confronted that problem, let me know, because I think it'd be very valuable to be able to have that live kind of data shared between everyone.
But do you have a suggestion how to do that?
At the moment, the only way I've tried before is an email list server. So just sending an email shot out to all the people once you hear something different about your sharks.
Yeah.
And I can do that, but you get lots of emails. So
Yeah.
I don't know. I'll ask the people who are involved in the studbook.
Yeah. Yeah, maybe we can also do something via the website. We could ask Stefan. Yeah. Thank you. Daniel?
Yeah, I would be happy to hear about any good marking method for elasmobranchs in general. What is the state of the art? We need to identify our animals. And when you see it every day, of course, you can work by individual characteristics like dots or scars or whatever, but once you give this animal away, after 2 years, you're certainly not able to identify it anymore. Are there any recent advances in marking or whatever?
I've just seen in Atlanta, I've been in a raw at Atlanta, and the Americans do some tattoo. They have asked a guy from a tattoo shop to, dots, different colors, and that seems to be quite successful. I can't remember the name, but I can find out who it was and to ask what the experience. But it looked quite good, so they asked a local tattoo shop to mark the animals. And even good for education, because the people from the public could recognize the special fish for like a sea bass or something as well. So.
Thank you.
Have you ever been trying just microchips to monitor? I mean, those fish who are individually fed should be easily then identified by this little reading thing for the microchip.
Yeah, for the microchip you need to catch the thing, which is in certain occasions not easy.
Yeah, but you catch it for transport, then you have it in hand.
Just to add to that, we pit tag our black tips, which is, I think, what we're talking about. But the other thing, it's a bit like a lightsaber from Star Wars that you can use that actually goes in the water, so the divers can go down near the animal and just wave it above it, so you don't have to catch it. Or also, you can catch them in a net or something and do it quite near it, but it's not quite the next stage, which is, I suppose, having the pit tag reader the other side of your aquarium glass, and so every time the shark goes past, it comes up with the number. That's something that we've been looking at.
Juan?
Yeah, a couple of years ago in the Dubai Mall, we had 47 tigers, so we had a problem. So we tried silver nitrate. That worked quite well. The problem is the public can see the numbers or the markings. The other one is a microchip. We found these readers from the pig industry. They're very cheap. That's the reason we got them. We did several tests to put in little housings, and in the end, it works perfectly. You had tag the animal in transportation, divers go in, pass the machine next to them, and you can read it fairly easy. If you drown the machine, it costs you $50.
How long does silver nitrate stay on?
It's for a couple of months. But we used to get every animal, probably every month, to get blood samples. So every month we just redo the marking again.
It depends on the species as well. You wouldn't ever silver nitrate a Calmatic ray or an eagle ray, but carcharinids are up to 6 months sometimes, or up to a year. It depends on the animal and its growth rate, really. If they're mature breeders, they should last for 6 to 12 months with silver nitrate, really properly applied silver nitrate dots on the pectoral fin.
Yeah, and there is no chance of inflammation because it's kind of burning. You do or not?
Yeah, generally, that's basically what you're doing. You're burning it. Our method was always to dry the area, dip the silver nitrate in fresh water, apply it, and then gently rinse salt water over it, and it basically goes from a very light white mark to a dark gray spot. So yeah, it's just a very tiny mark, and we had never had an infection or disease issue with it. However, I have heard stories of adverse reactions with rhinobathins and other soft-skinned, scaleless rays. So I wouldn't recommend it for those.
Okay. Yeah. Thank you. I would like to go on. Thank you very much for your presentation. Just very shortly about the elasmobranch part, what next? Well, in any case, I hope in 2013 and 2014 the coordinators will start to ask you a lot of information. Please give your information. But this is also important. So we have some vacancies. When making this list, I heard already Brian saying, well, Rachel maybe wants to do the Motoro. So it's really nice that this list already becoming a little bit smaller. But we will put this list on the website. There are in any case still some species open for programs, for coordinators. So if anybody is interested, please contact me as soon as possible. Well, I think we had the discussion already. Also looking at the time, I think we should proceed. I want to have, during the lunch, for all the coordinators, I would like to sit on one table and to hear a little bit how it's all going and if there are any problems. So please, for all the coordinators, let's try to sit on one table. Thank you. Brian?
Hello. Hello. Okay.
Now it's a 2-hour.
Sorry, it's just a very quick announcement. Before Dennis at the back there was talking about the idea of kind of like a preferred list of suppliers, and the Americans, colleagues, have been getting on with this, and I think they've got quite far with it. And previously at the collection plan meeting we had, we were also talking about the idea of some kind of supplier evaluation or some kind of preferred list as well. So anyone who is interested in discussing this and seeing if there's some kind of way forward we can go with a collaboration of aquariums, we wondered if maybe we could meet at the end of today after the last session. So anyone who's interested, just come to that session and we'll discuss if there's anywhere we can go with it and have a look at what Dennis has been doing. So thank you.
Thanks, Chris. There is one last presentation. We've been talking about sharks and rays for a while, but Dirk sent through an update on the Seacor thing. It's only a few slides. It was originally a lot of slides, and I said make it smaller. And Andrew's kindly agreed to give the presentation on Dirk's behalf. It shouldn't take very long, but there we go. Do you want that?
No. Yes, it is. It's quite brief. So, Seacor, the mission is to promote coral reef conservation through research, education, outreach and restoration, as I'm sure most of you are aware already. This is a quick sort of update of where and what they've been up to over the last year or so. The project in Curacao, that's been in partnership with Penn State University, the Aquarium in Curacao, Columbus Zoo, the University of Amsterdam, and the Caribbean Research Management and Biodiversity Foundation. The main impetus really of the projects that have been set up around Seacor is to go out into the areas and try to address, particularly with this one, it's Caribbean corals that are endangered within the environment. This one is focusing on Palmata and Cervicornis. The key to the Seacor really is about training workshops. It's about getting as many people together as possible. So whether that's local stakeholders, public aquariums, scientists, to try and bring together a knowledge base that they all bring a little bit of something to it, but they also get a lot out of it as well. The workshops that have taken place over the last sort of 11 years have included roughly around about 47 different institutions. So increasing that knowledge base for coral conservation and research, but also bringing to it, particularly from our sort of industry, it's the knowledge and understanding behind the system development for the actual care of the practice itself. If you look in the bottom right-hand picture there, that's Curacao, and it gives you an idea of sort of one of the things that often public aquariums bring to the fieldwork is the ability to develop systems for looking after the corals themselves. Going out into the field, collecting the coral spawn, bringing it back into the lab-type environment, getting good genetic viability and fertilization, growing on the planula larvae and then being able to settle it out to become that primary polyp and then growing it on again. There's a great deal of expertise over the last sort of 11 years to hone this practice and get it better and better and better. to the point where the Curaçao, they have a PhD research project that's been looking at exactly this practice and getting it as good as possible. So far, with some coral spawn that was collected in 2010 with the palmata and initially cervicornis, but unfortunately in the subsequent years, they haven't had such good spawning. So that's meant that as a species that's directly underneath the research, they've left that to one side and focused more on the palmata. So with developing these techniques and particularly the system designs behind being able to raise them on, they've been able to get roughly around about an 80% success rate in recruitment. So that's down to from fertilization through to that primary polyp, which is really, really good and significantly better than anything you'd find in the wild normally. Then, if you see in the top right there, that's after about 12 months of growing on in the labs in Curaçao, then those are transplanted back out onto the reef themselves. And it's fantastic to see that after 24 months, they had 100% survivorship on all the frags that were transplanted back out onto the reef. So that's where sort of Curaçao's been up to. They've been expanding further and further around the world. And this was in Guam. The workshop took place just in August time. It had, again, the partners were Henry Dorley, Pittsburgh Zoo, University of Guam, Guam government, and Underwater World, which is actually where the fieldwork took place. And where, again, as a longer-term project, they're building facilities there so that each year they can go out, collect the coral spawn, bring them back into the labs, grow them on, and then transplant them back out onto the reef. We actually had one of our coral biologists went on this trip, and again, from our point of view, I mean, we have some skilled coral husbandry within our institutions, but actually being able to take them out and put them into a workshop with lots of people from all around the world, both aquarists and stakeholders generally, they're able to get so much more information out of it, and they come back with much more a better ability of an understanding of the corals themselves, both directly with the skills that they've learned from being able to go out onto the reef, collect the coral spawn, bringing them back, but developing the systems too, so practical knowledge increases as well. So it's kind of a win-win from our point of view to send the aquarists out there, get the skill base increasing, which they always come back and apply back into your institutions too. There was 35 participants in this 10-day workshop, which was from 6 different countries, and they focused generally on Humilis and Tenui. The last one was Mexico, and this one, again, was in partnership with National Marine Parks, University, Mexico, Aquarium, and Pittsburgh Zoo. Similar sort of setup again, and it's aimed at being a longer-term thing, so they're developing systems, setting them up there longer term, have more and more people from around the world being able to come into different environments. And then with this wider sort of branching network, going around to different areas and different seas, you get to see a lot more and increase your knowledge base of the different environments where the corals are specifically from. But this one particularly was more aimed at stakeholders rather than the aquarium affiliates this year. Other things for this year then, we have a new website. Please go have a look at it. It has a lot more information on it about coral biology generally, but also the work that CCAR gets up to over the last sort of few years and going forward. And last and not least, having, for us, for example, having sent out an aquarist out into the field and come back into our aquarium, we have large coral exhibits as well, putting in some information about coral biology and coral conservation isn't a new thing, but actually being able to link it directly through into CCAR, and then being able to show that maybe the footage of your aquarist that's been out there is really, really useful as a great tool to engage more of the public, and that's exactly what the Horniman have done this year too. I think that's us. If you want any more information, contact Dirk, the slightly taller, more German version.
Thanks, Andrew. Okay, I won't keep you any longer. I just want to say thanks for all of you that came today, and hopefully you found it interesting, and hopefully it's generated enough interest to get much more people involved in participating in all of these really, really good programs. Thank you very much.